Macromolecules. Cells join small organic molecules together to form larger macromolecules. Macro means large. They may be made of thousands of atoms. They may weigh over 100,000 Daltons. And the four major ones are carbohydrates, lipids, proteins, and nucleic acids. And each cell has different macromolecules. Rather different quantities of macromolecules. Thousands of them. They vary among cells and individuals and even species. Macromolecules are made of smaller molecules, which are collectively known as monomers. And they can be joined together in different lengths. And with various configurations. Carbohydrates. Carbohydrates are also called sugars. Colloquially. They can be simple or complex. The simple ones are known as monosaccharides. There are also disaccharides. They consist of two monosaccharides linked together, and there are polysaccharides, which are many monosaccharides linked together. And they are the most numerous molecules of life. Let's take a look at some. Ooh, delicious. Loads of carbohydrates in your foods. Monosaccharides, they have molecular formulas that are submultiple of CH2O. So CH2O to the N. Monosaccharides are classified by the number of carbons in the backbone. So triose sugars have 3. Pentose sugars have 5. Hexose sugars have 6. And one thing you'll notice here, there are 2 hexose sugars. What is the difference between them? Glucose and galactose. They're both 6 carbon. Katie? Yes, you do need to know the formulas of sugars. You don't need to know their spatial arrangements. You will never need to draw them as such.
Okay, on the third carbon from the bottom, the OH and the H are replaced.
They're replaced, aren't they? Yeah, so that gives them different properties. That glucose tends to be sweeter than galactose. Fructose has the same molecular formula. It's also slightly different configuration, and it's sweeter than all 3 of them, all 2 of them. It's the sweetest one of the 3, fructose. And what is the function of monosaccharides? Here's fructose here. One major difference, double bond to that carbon. Function, major fuel for work, for cellular work. And they are the raw material for other molecules like amino acids and fatty acids. the ones that we synthesize. We don't synthesize all the amino acids. And the fatty acids, also the ones that we synthesize, we don't synthesize them all. So monosaccharides, oligo or disaccharides. Oligo means many, di means two. Joining two monosaccharides, usually in an aqueous solution, is due to a reaction known as a dehydration synthesis. So dehydration synthesis, which requires energy as it's a synthesis, results in the loss of one water molecule, resulting in the two monosaccharides being attached by an oxygen, one oxygen. What are our various, where did it go? What are our various disaccharides? Sucrose, of course, is one. That's your common table sugar, is sucrose, and that happens to be a glucose and a fructose together. Glucose and fructose. A disaccharide. Another disaccharide is maltose. And maltose is acquired by joining two glucose molecules together. Maltose. And the other one is, what is the other one? I forget. There's one more. Sucrose, maltose, lactose. Lactose, that's the other one, which is a glucose and a galactose. This one. So galactose derived from milk, maltose derived from malt, fructose derived from fruit and other plants. And of course, glucose is the molecule formed from photosynthesis. So that is the beginning of everything. Polysaccharides, hundreds to thousands of monosaccharides joined by glycosidic linkages. Those are the bonds that attach the two monomers of carbohydrate. One function is to store energy. Some are building materials. Starch is one you're familiar with. A storage polysaccharide. It's all glucose. It's just simply glucose monomers. It's found in plants and they use it to store energy. It's kind of like a plant fat. Well, when we or something else eats the plant, we get energy from the starch. Animals also store glucose, but that polysaccharide is called glycogen. Glycogen. There's starch, and starch has a 1,4 linkage of alpha glucose monomers. What the 1,4 linkage means is that whenever you see a diagram like this, a ring, sometimes, of course, there will be a O for oxygen, but if there's nothing, like here, 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 and here, we know that those are carbons. And carbons in a ring are numbered. And the number 1 is always the right-hand carbon in that ring. So that's 1, 2, 3, 4, 5. So it's the 1 linkage to the 4. Excellent. Cellulose is a 1,4 linkage, but of beta glucose monomers. So you notice that the difference is the placement of the oxygen. It has two slightly different placements, so that causes it to form a different kind of bond. Indeed, it's a very strong bond, and its function is not that of energy, but rather that of structure because of its strength. So starch is all glucose monomers, and as is cellulose. This is an example of cellulose. And because of the configuration of the monomers, cellulose forms hydrogen bonds between the chains, and they're parallel. And parallel structures tend to be very strong. So tendons and ligaments, for example, that you have are quite strong because the fibers are parallel. But these are cellulose molecules, and you find them in plants, for example, in the plant cell wall. It's stored, sorry, not stored. Strike that. Glycogen is stored in the liver and muscle, but there's only about a day's worth. If you don't eat for a day, then you've used up your carbohydrates. Oh, one thing I did want to say about cellulose. Cellulose is what is known as insoluble. So as it passes through the intestine, it abrades the intestinal walls, causing mucus to occur, and it helps food to pass through the digestive tract. We cannot digest it for its nutrients. Cows can, but we cannot. So you notice the difference between cellulose. Look at the cellulose structure, and then look at the glycogen structure. It's quite different, isn't it? It's very branched. And another important polysaccharide that you don't talk about in anatomy and physiology, because nobody really cares about chitin there, but chitin, it's a very important structural polysaccharide. It's used in the exoskeletons of arthropods, insects, spiders, and crustaceans. It's very similar to cellulose, but it contains a nitrogen appendage. So similar to cellulose, but with a nitrogen containing appendage. And usually chitin is a leather, it's got a leather-like feel to it. It's a bit flexible, except when fortified by calcium carbonate. And then it becomes a hard shell. And you find chitin also in the walls of fungi. Lipids, fats. Lipids have 1 to 3 fatty acids attached to glycerol. There are saturated fats, there are unsaturated fats. The difference is that the unsaturated fats have double bonds. Triglycerides are the most common lipids, best energy source. Phospholipids have 2 fatty acid tails and a polar head. They make up our cell membranes. Sterols, another very important lipid, have no fatty acids. Cholesterol, for example, is a sterol. And they're membrane components. And also, cholesterol can be converted to many other things: vitamin D, steroids, bile, and salts. Lipids are highly diverse in form and function. So yeah, lipids don't really have monomers. They don't form polymers. They form a sort of a polymer, but not really considered a polymer. Where do you find lipids? Ooh, you find them in your fats, in animals, which tends to be saturated, your liquids in plants, which are unsaturated. So butter, which is made from milk, is saturated and solid at room temperature, whereas plant oil is liquid at room temperature. The kind of unifying feature of all lipids is that they have no affinity for water. No affinity for water. They're hydrophobic, and that's because their bonds that they form, CH bonds and CC bonds, they're just simply non-polar. Because of non-polar bonds. So here's the formation of a triglyceride. This one is called triacylglycerol. So this is your glycerol group here with some hydroxyls. And this is your fatty acid. This one happens to be palmitic acid. It's a straight chain. It has a carboxyl group at one end, which easily dissociates when synthesized into a water. Fairly easily, but it's not spontaneous. Of course, it's a synthesis, which always requires energy and ATP. So why are they saturated and unsaturated? Well, a saturated fat has all hydrogens. Possible. But an unsaturated fat has this double bond here, and strangely, that makes it bend. And when it's bent, it can't form a solid structure because it just tends to have space in between the molecules and it can move around those molecules. So it's a liquid. The function Of fat are many. One is energy storage. There's quite a lot of energy in fat, more so than in polysaccharides, interestingly. To cushion vital organs. And to serve as insulation from cold. So we were talking about this the other day, weren't we? Yeah, Eduardo asked a question. So marine mammals have a very thick layer of fat. Another type of lipid is called a phospholipid. It doesn't have three, it's not a triglyceride. It has two fatty acids. And interestingly, one of them is bent. So what does that tell you about the cell membrane? So all of our cells, as indeed all cells in the universe, consist of a plasma membrane that contains the organelles inside. And it's a combination of this hydrophilic head here, that's these guys, and the hydrophobic tails here. So by looking at this, why is this hydrophilic? We've got a choline, a phosphate, and a glycerol. What makes it hydrophilic? Why does it readily dissolve in water?
Hydrogen bonds?
Because of that negative
No.
charge on oxygen?
The negative charge, yeah, that's it. This is a negative charge. Yeah. The other bonds are actually non-polar. So it's just that negative charge on the phosphate group. Good, 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 good. So what's really interesting is you put a bunch of phospholipids in water and they'll self-assemble into this double membrane with the hydrophobic ends facing each other and the hydrophilic parts facing the outside. That's very handy. So it forms a barrier between the inside and the outside. Steroids, that's another kind of lipid. They have a carbon skeleton consisting of 4 fused carbon rings. They always do. That's a steroid. They all come from cholesterol. So cholesterol is the precursor from which all other steroids are synthesized. So they all have 4 rings. So you should be able to recognize a steroid. 4 rings. I wouldn't ask you to say, is this an estrogen or is this a testosterone? But to recognize it as a steroid. And of course, cholesterol is essential, but an overabundance of cholesterol can contribute to cardiovascular disease. Proteins. Some people say we're carbon-based. Some people say we're protein-based. Well, we are both, of course. Proteins are influential in about everything that an organism does. It includes storage, structure, transport, signaling, movement, defense, and enzymes. So they regulate metabolism. They selectively accelerate chemical reactions. We have tens of thousands of various proteins, each with its own structure and function. They're the most complex molecules known. They're made of polypeptides, and polypeptides are polymers of amino acids, and there are 20 amino acids. And this is what they look like. Here's a bunch. Here's some that are nonpolar, and here's some that are polar. So do you notice anything similar about these 3 amino acids here? What are the functional groups that you can see? What are the functional groups? What's this one called? An amine, good. And what's this one? It used to have the H there, but it doesn't anymore because it's dissolved.
Is it carboxyl group?
Carboxyl? Marcus.
Yes, sorry.
I didn't see it come in.
Sorry.
Carboxyl group, yes. And then the hydrogen, that's the same. So these 3 things are exactly the same for all amino acids. The only difference is this R group. It's called an R group. And that is what varies. And there's 20 of them. And it's the R group that gives amino acids their particular properties, such as being nonpolar or polar. So the physical and chemical characteristics of the R group determine the unique characteristics of a particular amino acid. Yeah, so some are polar, for example. Some are bases, or some are acids. So the amino acids are joined together when a dehydration, is always a dehydration reaction that joins everything together, removes a hydroxyl group from the carboxyl end of one amino acid and a hydrogen from the amino group of another. And the bond, remember the name of this bond, is peptide. It's a peptide bond. With sugars, it's a glycosidic linkage. With amino acids, it's a peptide bond. And the functions are numerous, and they totally and completely depend on shape. Like the hemoglobin we saw in the Nottingham professor's, I don't remember his name, fortunately, but remember he talked about hemoglobin, and hemoglobin's shape is what make it function so well as an oxygen transport. In almost every case, the function depends on the capacity to recognize and bind to some other molecule. So in the case of hemoglobin, it's the ability to bind to oxygen. So e.g., hemoglobin to oxygen. Enzymes recognize and bind to specific substrates and initiate a chemical reaction. Neurotransmitters, so lots of neurotransmitters are proteins, pass signals by binding to receptor sites on proteins. So how do we get this shape? How do we get the shape of the protein? It's very interesting. It starts with a primary structure. The primary structure is simply the order of amino acids. So here we've got valine, glycine, serine, and leucine, and then all the other ones that are there. So that's the primary structure. The secondary structure can be two different shapes and is caused by hydrogen bonds, forming very particularly depending on the sequence of amino acids. So caused by H bonds. So one structure may be a pleated sheet, a beta pleated sheet. So you can see the hydrogen bonds here between oxygen and hydrogen of the two strands of amino acids. Or it could be a helix. Here are the hydrogen bonds in the helix. So a long string of amino acids can form in the same polypeptide these two secondary structures. You put them together, when they start to fold, it's called a tertiary structure. So you can see that there's some helices there and there's some beta pleated sheets in the same protein structures. That's the tertiary structure. That is the shape of function. But sometimes there are more than one subunit, in which case it will be called a quaternary structure. Hemoglobin has a quaternary structure because there are 4 subunits of hemoglobin. So my question to you is, what determines the sequence of amino acids? What molecule determines that sequence? That we haven't talked about yet.
The mRNA?
Exactly. So DNA, which is transcribed into messenger RNA, and that will be read, and it will result in a particular sequence of amino acids. So it's really important. This sequence is really important. So here, for example, are red blood cells, normal red blood cells, and the primary structure of normal hemoglobin. Of course, it's just a section. Here, valine, histine, leucine, thrombine, proline, glutamate, possibly. But what have we here? On 6, oi, 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 we have a valine. Big mistake, because it's a genetic disorder, and it's caused by this one amino acid being in the wrong place at the wrong time. And what it does is it makes the hemoglobin clump together. So it makes it sticky. And when the hemoglobin is sticky, it causes the cells to form this shape like that. Sorry, it should be red, shouldn't it? It's like a sickle shape. That's why it's called sickle cell anemia. And those cells cannot transport oxygen. Yeah, so the structural properties of silk are due to beta pleated sheets. That makes silk fiber incredibly strong. So strong, it's just not funny. And I'm going to show you a video on that in a sec. But what's very interesting is that protein structure can change in response to physical and chemical conditions. We saw that with the hemoglobin, remember? So the oxygen comes along, binds to the iron fits into the heme ring, and that tugs on the rest of the molecule, and then the molecule has a greater affinity for oxygen. So it just changes a little bit in response to chemical conditions, which in this case will be the oxygen. But other changes like pH, salt concentration, temperature, can unravel or denature a protein. In some case, it might not go back, but in other cases, it does. It denatures and it renatures. So for example, in the case of, say this is a plasma membrane, and you may have a protein that's a channel in one shape, and then it will change its shape to allow molecules to pass through, but it will go from one shape to the other. How do we know the shapes of proteins? Well, you can use something known as X-ray crystallography or NMR, nuclear magnetic resonance, to determine protein conformation. So you basically crystallize the proteins, throw an X-ray beam through it, and then you can determine by electron densities what the shape of that protein is. Okay, 5:14. I'm going to stop there for now. Thank you for watching. We'll be back.